Thanks, Dan. Thanks for the introduction, and thanks for the opportunity to tell you a little bit about our research with C4 perennial grasses. So my background is in ecology and evolutionary biology, and my lab really has been interested in local adaptation in natural populations. So how does the interaction of natural selection, genetic drift, gene flow drive the evolution of traits that allow plants to have high performance in their local environments? And one of the interesting things about grasses is that we often see ecotypic divergence along lines associated with precipitation and rainfall. And so in many, many cases, we see the evolution of upland and lowland ecotypes of grasses. And so really, since I met Dan, we've been interested in trying to understand variation in switchgrass that occurs across North America that's associated with these upland and lowland ecotypes. And so here's just a picture, a nice representation of a lowland switchgrass that's highly productive, usually occurring in the south and associated with habitats that are seasonally wet. And next to that, I have a photo of an upland switchgrass that occurs in more xeric stressed habitats. And so we'd like to understand sort of the interplay of the sets of traits associated with uplands and lowlands and how that impacts performance across gradients like precipitation. So we'd like to understand this from a natural evolution perspective, but obviously this has pretty big implications for how well we could utilize switchgrass in terms of a biofuel feedstock. And one of the things that we're really interested in is to trying to understand trade-offs that might occur. So one idea is that if the plants evolve traits that work well in a mesic environment, that might come at some cost in terms of performance under a drought-stressed environment. And so we really want to try to find the genes that underlie these traits and actually try to understand how those genes drive trade-offs. And one of the ideas is if we can do that, then perhaps we can manipulate those genes in particular ways to reduce costs or maybe even breed generalists. And so we use many different approaches. We work mostly in the field. So we take these plants out in common gardens. We do reciprocal transplants. We do a lot of QTL mapping and association mapping. And if you're interested in those ideas, come to my poster tonight, poster 16, and I'll tell you a little bit more about that work. But what I wanted to focus the short talk tonight on was to tell you about a second system we're working on, which is Panicum Holly. So we started working with switchgrass 5 years ago, and it's been really an interesting experience for me to see how hard JGI has worked to create the resources that are the flagship genome of the Panicum virgatum genome. So it's a very, very complicated genome. It's a tetraploid. It's a large genome. Switchgrass is an obligate outcrosser, so the genome is highly heterozygous. There are many, many challenges to working with tetraploid switchgrass. And so along the way, we became interested in close relatives of switchgrass that were diploid, that had smaller genomes and might actually facilitate us to use a variety of different genetic and genomic tools in the context of understanding local adaptation. And so we've been working with Panicum Holly, and over the last few years, we've been able to generate a reference genome in collaboration with JGI, and that genome has helped in the assembly of the virgatum genome. And now we're sort of, in a comparative way, trying to understand the evolution of upland and lowland ecotypes in both of these perennial grass systems. And I really want to make a call out to the JGI staff that have helped, and in particular, Jeremy Schmutz and Jerry Jenkins, that have really worked hard on these assemblies and generating these tools that I think the community is just now beginning to leverage. So Panicum Holly is native to North America, just like switchgrass. It occurs across the southwest of the United States. There are actually 2 varieties or subspecies that correspond to very similar to switchgrass upland and lowland types. It's variety Holly that is the upland type, and variety Philippes, that's the lowland type. We primarily find Philippes growing on the coast, the Gulf Coast, in mesic depressions that are seasonally wet. And we find Holly occurring all across the Southwest in a whole variety of different habitats. It's a relatively small genome. It's diploid. And one great thing is we can go from seed to seed in a very short period of time, so about 6 to 8 weeks. So it's a tractable system. It's smaller than switchgrass, so we can grow it in the greenhouse, grow it in growth chambers for our experiments. And here's just a picture of sort of characteristic variation between these upland and lowland types. The lowland type flowers much later. It's larger in stature, has much thicker, longer, broader leaves and more tillers. Tends to be more of a solid perennial. In contrast, the Hollyi type, our upland type, is much more diminutive, has smaller leaves, smaller inflorescence, tends to go dormant during really hard droughts, and is much less of a solid perennial in many cases. And so we've been doing lots of field collecting, driving across the Southwest, finding new populations of switchgrass. Here's just some examples of Hollyi, some examples of Hollyi at different locations in Texas. And then we've been bringing that material back to Austin and growing them in common gardens to explore natural variation. And there's just an enormous amount of variation that we find in both the Philippi's and the Hollyi types. We're really interested in seed size evolution that might be related to establishment. We're interested in tillering and growth morphology. And most recently, I have a graduate student that's very interested in roots. And we really don't know a whole lot about perennial roots. So I think it's a really nice new opportunity. So I come from a genetics background. The first thing we do is make a cross. We took our 2 genome reference lines, made an F2 mapping population. We were able to leverage the genome resources that Jeremy and Jerry put together to map our RAD genotyping platform to generate a linkage map fairly quickly. And we found, I think, a pretty surprising result. So in our initial cross, we found that there were really hot spots in the genome where lots of the traits that underlie this divergence between upland and lowland map. So we found, this is just a representation of the Hollyi genome. There are 9 chromosomes. And we mapped these 2 fairly coarse QTL regions where virtually all of the traits we're interested in map. And that either suggests that there are some genes in tight linkage that underlie this ecotypic divergence, or maybe that there are some genes of major effect that are pleiotropic and impacting a whole host of these different traits. So a very exciting result. My student interested in roots just recently looked at roots, and sure enough, these QTL also affect root variation. So they may be some interesting master regulators. So another really important aspect of abiotic stress responses, very commonly observed, is that gene expression changes in response to stress underlie adaptation. So we've been really interested in looking at expression divergence between the upland and lowland types, and would love to know more about whether these differences are driven by cis variation or trans variation, as a number of the speakers earlier described. And so one of our most recent projects was basically looking at expression variation as a quantitative trait locus, thinking of expression as a trait that could be affected by the environment or by genes segregating underlying divergence between these 2 ecotypes. And so here's just an example of one of our F2 mapping populations planted in the field. We have the parents, F1 and progeny. We can clonally replicate these plants, which is another really nice feature for doing these types of studies. We let plants experience a natural drought stress, harvested tissue, and then rewatered everything to simulate a rainfall event, harvested tissue, extracted RNA, and then in collaboration with JGI through one of the community sequencing programs, did total RNA sequencing of this population. And the idea here was to do eQTL mapping on this ecotypic divergence. And so we see remarkable expression divergence between the upland and lowland types in this experiment. This is just comparing log fold change of the upland to the lowland under the recovery versus the upland to the lowland in the drought stress experiment. So we see a lot of genes that are drought responsive, and then we see, more interestingly, the red, blue, and green are genes that are either uniquely up or down regulated in one of the types, or actually that are up or down regulated in different directions between the 2 uplands and lowlands. So quite a lot of variation. And for us, the exciting thing is now that we have a genotype screen on this F2 mapping population, and when we compare it with our RNA-seq data, we can actually take this to a level of mapping QTL. And so I have two really fantastic postdocs in the lab right now, John Lovell and Scott Schwartz, that have been developing an eQTL pipeline. This allows us to scan the genome for QTL that affect expression, that affect expression in response to environmental variation. So QTL by environment interactions. And what's, again, surprising to me is we actually see highly heritable expression variation. Probably 2/3 of the genes appear to have some heritable variation in this mapping population, with some of them having really, really high heritabilities, so very strongly genetically controlled. Overall, we see a real predominance of cis variation. We map very strong, very solid cis eQTL for probably half of the genes. And in fact, so many of them are strong cis eQTLs that it's made the QTL mapping actually difficult, because what we need to do is control for these big effect QTL first, and then scan for more minor effect QTL across the genome. So clearly, expression variation is rapidly evolving in these ecotypes. And then this is sort of a global plot of our eQTL study. So on the x-axis here, we have a representation of the 9 chromosomes and the position of QTL that we map. And then on the y-axis here is the position of genes that are the targets or traits we're studying, the transcripts in the genome. And each dot corresponds to a QTL. And the red dots correspond to QTL that are constitutively affecting expression. And the blue dots correspond to QTL that are interacting with the environment in differential ways between the upland and lowland ecotypes to control expression. And what you can see is on this axis, the diagonal here, a really dense set of spots. Those are all representing the cis eQTL that I mentioned that are very, very strong effects in the mapping population. But what's really interesting and exciting is we also map a transband. So a transband would be a piece of the genome that's having an effect on many, many, many different target transcripts. When we look at this transband, this is in the same position as one of the QTL hotspots that we identified for all of our above-ground morphological traits. So it seems that these hotspots are not only affecting growth and morphology and flowering time and physiology at a whole plant level, they're also driving lots of variation at the expression level. And then secondly, the genes, the transcripts that fall in this transband are enriched for environmental interactions with the drought stress. So we're just finishing this analysis now and trying to write up this paper. One exciting thing is if we take the genes in this transband and do enrichment analysis, we see some interesting Go categories that pop out as enriched. And we also see some promoter motifs that are enriched in the targets. And so a big part of my lab, probably for the next 3 or 4 years, will be trying to find, map, and ultimately clone this transband regulatory QTL. So hopefully in a couple of years, I'll be grayer, but I'll come back and I'll have maybe a molecular story to tell you all about Panicum Holly. So just to end, Panicum Holly is in a great situation right now. We have a really solid genome. We have lots of collections of natural diversity. We've resequenced a lot of that material. We're hoping to do GWAS analyses in the future. We're working on transformation protocols. So if you're interested, come to poster 16. I'd love to tell you a little bit more about the resources and how I hope it can help impact our understanding of these C4 perennial grasses. And then I'll just end by saying I think we have this really interesting, unique case where maybe the architecture, the genetic architecture underlying this divergence is actually relatively simple. That may be a unique case. And then just to mention a few people that have been involved in these projects, David Lowry was a postdoc in the lab. David was really interested in learning about C4 perennial grasses and did a lot of field work and a lot of collecting, made many of our initial crosses. He's now started an assistant professor position at Michigan State and continues to work with the grasses. As I mentioned, Jeremy and Jerry have been really important. Avinash, Shu, Dan, Carrie, the whole JGI crew has really been important in developing the system for these grasses. In my lab, Scott and John have really led a lot of the eQTL mapping. Juan Diego has been in the field traveling and doing lots of collecting and Albina is interested in roots and starting to do our root biology. So thank you for the attention, and I'd love any questions.
Thanks, Tom. I think we have time for one question. I always have questions for Tom, so anybody else? Get a mic from the back.
Yeah, so for the transband, do you have any idea how many genes are involved there? Could it just be a broad QTL because of, mapping error? Or do you think that it's multiple?
Yeah, for sure.
loci?
It could be. Again, even if it's a broad QTL, having all of these traits mapped to the same location suggests to me it's a pretty interesting area no matter what. There are a lot of commonalities in these traits. I don't think it's unreasonable to imagine a meristem regulating gene or a flowering time gene having a whole series of cascading effects like this. That would be a nice case in terms of us making quick progress. So we have a recombinant inbred mapping population that will replicate a lot of this in, and have made lots more recombinants. So we'll know more soon.
Thanks again, Tom.
Okay.